Hi everybody, my name is Cameron and today we're going to be looking at genetics and reproduction, specifically sexual versus asexual, DNA, its structure and its function, as well as inheritance. Okay, so when it comes to asexual reproduction requires a single parent. Okay, the offspring are going to be genetically identical to that parent. Okay, we call them clones. The common methods that are binary fission within bacteria, budding in some hydra are like little seaweed creatures, they have budding, fragmentation such as a starfish. If you cut the arm off a starfish, not only does the starfish grow the arm back, but that arm grows into another starfish. And spore formation, so that's in your mushrooms or your fungi. Okay. Some of the benefits is that it's incredibly fast. You don't have to find a mate. It's just done by that organism on its own. And all the positive genetic characteristics of the parent are passed down to the offspring. So you don't have to worry about another individual's characteristics dominating them. Okay. Some of the negatives is that also the negative characteristics are going to definitely be passed down to the offspring. Okay. So if you have a crop and that crop are all clones of one another and a disease comes in and the one individual that all the clones are from is not immune to that disease, your entire crop's going to be wiped out by that disease due to the lack of genetic variation and the lack of possible mutations that would have occurred that may have made a couple individuals resistant to that disease. When it comes to sexual reproduction, this involves the fusion of haploid gametes. So that means it requires two parents. The offspring are going to inherit characteristics from both parents and they're going to have this genetic variation. It's also important that these gametes that are created have variation between them. If a mother and a father have three sons, okay, three boys, those boys are going to look similar to each other, but they will be different, even though they are from the same parents and they are the same gender. They'll still look different because each gamete is going to have this genetic variation. So how these gametes are formed is through a process of meiosis. Okay, we'll go through that in later slides. It happens in animals as well as flowering plants. And some of the benefits is that it creates that genetic variation that we talked about. And this also allows for natural selection to choose characteristics that are most beneficial for that individual in its environment. Okay, so when there is variation, the stronger, the more fit characteristic will thrive. Okay, and the negatives is that it takes time. Okay, some animals have to wait for mating season where the females can't even mate before that mating season. Another one is like the anglerfish that lives very, very deep in the water, in the oceans. Okay, it's pitch black down there. And the male anglerfish is incredibly small. So he will spend most of his life eating and looking for a mate in the darkness. Okay, so that takes a while. If it was asexual, he would have just reproduced by himself, with himself, and made more little anglerfish. Okay, but he has to go find a mate now. It gets even harder in certain animals where they have to fight for a mate. Okay. Now, when it comes to sexual reproduction, it requires two partners and the joining, the fertilization of a female egg with a male sperm. Okay, so for that joining to occur and for them to share their genetics, okay, each gamete, each sex cell, okay, has to have half the number of chromosomes that a normal cell would. Okay, to get this, the cell is going to go through something called meiosis. Now, meiosis is the division of a cell, okay, into four genetically different sex cells or gametes, each of which is going to have half the number of chromosomes of the original cell. Okay. Now, if you don't see why it's 4, you can see down there. What happens is it initially goes through a normal mitosis sort of division, same thing, ipmat, okay? It goes through prophase, metaphase, anaphase, telophase, but then it goes through it again, the second sort of mitosis, okay? We call it prophase 2, metaphase 2, anaphase 2, okay? But all you need to know is that these gametes are haploid cells, half the number of chromosomes, and they are genetically different from one another. These are going to fertilize to form something called a zygote. This zygote is then going to grow through mitosis. Okay, this zygote is also going to be called a diploid cell. Diploid because it has the full number of chromosomes. Now, each species of animal is going to have a different number of chromosomes. So how we represent their diploid and their haploid is by using an N, okay, N for haploid, and 2N for diploid. Okay, so a zygote has 2N chromosomes, while a gamete has N chromosomes. So reproduction also occurs obviously in non-animal species or non-animal organisms. Okay, so in fungi it's sexual reproduction by producing spores and non-sexual reproduction through mitosis. Okay, so fungi have a mycelium network, and if a part of that mycelium network breaks off, it's kind of like a root network. And if part of that root network breaks off, it will form its own colony of fungi. Okay, so it's going to be genetically identical. It's asexual reproduction, but they also sexually reproduce to create spores. Okay, now in plants they reproduce asexually through something called runners. Runners are these stems that go horizontally across the ground, and they will root themselves into the ground. So even if it is cut off at the source, okay, this runner will still grow to be its own organism. Okay. They also produce sexually through pollination. Okay. And we call that germination. Then in something like malaria parasite, Plasmodesmata or something like that, it's called a protist, and it also reproduces sexually initially, okay, within the mosquito, but then once they get into the human, they reproduce asexually. Okay, so let's look at genetic information now. When we talk about genetic information, we're talking about DNA, deoxyribonucleic acids. Okay, so DNA is going to be a carrier of all the instructions necessary for a cell to function, for a cell to grow, for a cell to divide. All that information is carried inside of the DNA. Inevitably, that DNA is going to encode for a specific protein. Okay, so a certain section that we call the gene is going to encode for a specific protein. And we'll get to that when we do protein synthesis. Now, DNA is split up into nucleotides. Okay, now a nucleotide is made up of 3 parts. It's made up of a phosphate backbone, a sugar, and a nitrogen base. Okay, now there are 4 nitrogen bases and therefore 4 nucleotides. Okay, adenine, thymine, guanine, and cytosine. I know people say that differently sometimes, but that's how I pronounce it. So A binds with T, G binds with C. They bind with each other through hydrogen bonds. Okay, so that's why we have a double helix structure, meaning that there are 2 sets of nucleotides together and they bind with one another, complementary. Okay. Yeah. So the human genome, why we would study someone's entire genome, okay, is to look for genetic diseases. because in the future we might be able to genetically modify people to not have their diseases. Okay. It also allows us to understand how humans evolved, as well as their evolution, okay, and their history. And then, yeah, so in DNA, you're also going to have something called coding DNA, which is DNA that codes for that specific protein. Then you get some parts of the DNA called non-coding DNA. Now, non-coding DNA for the longest time was called junk DNA. They didn't realize, they don't understand, why is this DNA not being coded for a protein? Okay, but it turns out that that DNA is then going to control whether or not some proteins are made at certain times. You see, so you say it turns on or off the gene of that specific non-coding DNA. You see, it gets very detailed and quite intense, but you don't have to know that for now. You just got to know that non-coding DNA is going to turn on or turn off specific genes, while coding DNA is going to code for a protein. So this is just a diagram to show what DNA actually looks like. So again, we're going to have our nucleotides, okay, made up of a phosphate, a sugar, and then a nitrogenous base. Okay, so this over here is one nucleotide. This over here is another nucleotide. And as you can see, the phosphates are going to join to a sugar. And that makes up this phosphate backbone that we see. Okay, and we know that there are 4 nitrogenous bases. Okay. Apologies, my hair is now under control and I can carry on with this. Okay, so when it comes to DNA now, how exactly does it control all the processes of the cell? Well, it's going to encode for proteins. We call this protein synthesis. Now, the DNA itself in eukaryotes has to stay within the nucleus. It's too big to leave it. So what it's going to do is it's going to send a messenger. Okay. This messenger is going to make a copy of the DNA sequence for a specific protein. Okay. Now, this messenger is called literally messenger RNA. RNA because it is a single strand of RNA. Okay. Now, the difference between DNA and RNA, RNA is single-stranded, and instead of using thymine, it uses uracil. That's the only difference. Okay. So mRNA is going to make a copy, a template of our DNA. It is then going to be processed, which you'll go through later on, but it'll be processed and it'll be sent out the nucleus. Once it's out the nucleus, it's going to meet up and bind to a ribosome. Now, the ribosome is going to be sort of the factory of the protein. The factory where the instructions come in and then the protein is made. Now, these instructions are going to be read 3 nucleotides at a time. These 3 nucleotides are called codons. Okay, so on that mRNA, it reads 3 nucleotides at a time. Each codon is then going to be paired, complementary base pairing, with an anticodon. Okay, now proteins are made up of amino acids. They're going to be put in a certain sequence so that they can fold into a specific protein. But how do these amino acids get placed in this specific sequence? Okay, they don't just attach themselves to the mRNA. They use something called tRNA, transport RNA. The transport RNA is going to take that amino acid, because each tRNA is going to have its 3 nucleotides that are complementary to the mRNA's nucleotides. Okay, so it's called a codon on the mRNA and an anticodon on the tRNA. This tRNA is going to meet up with the mRNA. They're going to bind complementarily in the ribosome now. Okay. Then next to it, the next codon is going to receive its tRNA partner. Now, the amino acids linked to these tRNAs that are next to each other are going to join up. A long chain of amino acids is going to form as the new tRNAs come in and the old ones leave. And they leave without having their initial mRNAs on them. Okay. And yeah, as these amino acids are going to link to form an amino acid chain, it is then going to get longer, and eventually it reaches something called a stop codon. And that does not code for anything. The amino acid chain gets released out of the ribosome, and because of the certain amino acids and the sequence that these amino acids are in, they are going to fold into a 3D structure now. Instead of just a line, it's going to fold into a 3D protein. And that protein is then going to be able to perform its function. Okay. And that's protein synthesis. So we've heard about mutations before. Mutations happen in your DNA. Okay, but how exactly is that going to affect the rest of your body? So, as we know, DNA is going to encode for certain proteins. It uses its specific nucleotide sequence to encode for a specific protein. Now, if there's a mutation in that nucleotide sequence, if one of those bases, those nitrogenous bases, were to swap for a different base, that means there would be a different amino acid paired with that base. And that will result in a different sequence in the chain of amino acids, which will then lead to a different protein forming or a non-functioning protein forming. It can also happen in non-coding DNA, in which case certain genes just won't be turned on at all. Certain genes aren't going to be expressed. Okay. Now there are three different ways that the mutations can change the DNA. We can have one where a base is inserted in between two other bases. We can have when one is deleted, so when one base is just taken out. And we can also have one where one is substituted or replaced. Okay. Now, and you'll learn this later on, but each codon, because remember, it's 3 nucleotide bases that encode for one protein, one amino acid. Each codon does not encode for only one amino acid. Okay, it does. But there's some codons, you could have like 3 different codons that encode for one amino acid. Okay, so sometimes a mutation can occur and the same amino acid will still be encoded for. Okay, but if that amino acid, if the base had to change to something else, then a different amino acid could be encoded. Okay, so what I'm saying is some mutations don't affect the protein created and others do. Okay, and yeah, usually these mutations are harmless, they go undetected, but sometimes it can make either advantageous mutations or disadvantage mutations. Okay, yeah. Okay, now when it comes to chromosomes and genes, we know that DNA is extremely long. It's going to have to be packaged so that it is more handleable when the cell needs to divide. Okay, it can't just be a string of DNA. So the cell packages its DNA into something called chromosomes. So it starts by winding the DNA around proteins called histone proteins. Those histone proteins are then going to join together to form a chromosome. Now humans have 46 chromosomes and they pair up to form 23 chromosome pairs. Okay. Now in your normal cell, in a normal body cell, we're going to have 46 chromosomes. We call that a diploid. Okay. Now in those chromosomes, there are going to be sections of DNA that encode for a certain protein. These are called genes. Okay. So genes are sections of chromosomes that encode for a specific protein or trait. Okay. Now diploid, meaning 2. Okay. So you're going to get 1/2 of your chromosomes, and therefore 1/2 of your traits, from your mother, and then another half from the father. Okay, so that means there are 2 alleles per trait. Now, what is an allele? An allele is a variation of a gene. So something like hair color, eye color, how well your body produces amylase, if your body produces insulin at all, okay, that comes from your parents, and there are 2 alleles that will control every trait in your body, okay. And we have a dominant allele as well as a recessive allele, and we'll get into detail about that later. So here's just a little diagram that I drew to sort of show you how DNA is packaged. It starts off as that long strand of DNA, twists around histone proteins, which are then further going to group together to form a chromosome. Now, we have 46 chromosomes. Sometimes they will refer to them as chromatids. Now, in my mind, and what I studied at university, it takes 2 chromatids to form a chromosome. Okay? If it just helps you understand this a bit better, because they're saying we have, so 23 chromosome pairs. So a chromatid, chromosome, it's kind of used interchangeably. Okay? But this is how I sort of remembered it and how it helped me understand this, because when you go into meiosis, it gets a bit complicated as to when it's a chromosome and when it's a chromatid. So humans are going to have 2n number of chromosomes in your normal diploid cells. 2n, a full set of chromosomes. Okay, so when it comes to inheritance, where you get your traits from, okay, which parent, we know that you're going to get half your chromosomes from one parent and half from the other parent. So that means that for one trait, it's going to be controlled by 2 alleles. Now, you get dominant and recessive alleles, okay? And depending on the configuration, we call it different things. So if you have a dominant allele from one parent and a recessive from another, we call this heterozygous. Zygous, hetero meaning different, zygous meaning a zygote, because that's what's formed when your 2 gametes are going to fertilize and come together. They're going to form a zygote. So heterozygote or heterozygous is going to be a zygote with 2 different alleles for the same trait, for the one trait, okay? If it is 2 of the same allele, such as capital B or small b, dominant and a dominant, or a recessive and a recessive, we call this homozygous. Homo meaning the same, zygous meaning the zygotes, dominant or homozygous recessive. Easy enough. Okay, now when it comes to the dominant, so when it comes to explaining which part we're looking at, we use the words genotype and phenotype. Now, genotype is what allele you're going to have. Okay, so if you have a homozygous dominant allele, that is your genotype. It is capital B, genotype. Okay, now what that encodes for, what that shows as in the individual, is called the phenotype. You can remember this because pheno, face. I can't see your genes, but I can see your face. Okay, that's what your genes show as. They show as your face. Whereas genotype, we're talking about the actual genes that we can't see. Okay. So if we have a dominant gene, okay, a heterozygous or homozygous dominant, the dominant trait is going to show. If we want our recessive trait to show, it has to be homozygous recessive. That's the only way that the recessive phenotype is going to show. Okay, the best way to learn these words is to use them often. Okay, so in all of your questions, make sure you refer to the homozygous, you can even say the homozygous diploid, or you can say the heterozygous. Use the words the dominant allele, not the dominant trait sort of thing. Okay, and yeah, just practice. When it comes to Punnett squares, Same thing. So Punnett square is used to show the probability of the offspring having a certain genotype. Okay. Because there is only two alleles from each parent, and the possibility, there are only 4 possibilities for a specific trait. We're going to put the female usually on the left and the male on top, and we're going to cross them. Okay, so as you can see here, it crosses big B to big B, big B to small b, or big B, small b. Okay. And yes, this is going, so the genotype is going to be something like homozygous dominant. The phenotype is going to be pink. The genotype here is going to be heterozygous, but the phenotype is going to be pink. Okay. Now, other than a Punnett square, we can use something called a pedigree chart. Now, the pedigree chart is going to allow us to see the history of a family with regards to a certain trait. Okay, who's affected, who might have been a carrier, who was unaffected. Okay, and it doesn't usually indicate your heterozygous, homozygous individuals. It is mainly going to indicate affected, not affected. That's all. Okay, and this just comes with practice. So if you don't understand these, it's going to take practice. Make a session with one of us here at Fame Education, and we can teach you how to do this, and we can go over practice with it. But it really just takes practice. Okay. The males are going to be represented by squares, the females represented by circles. Usually people affected are going to be shaded in, colored in, okay, and people unaffected are usually just blank. The grayness, okay, not always, because in that case what they're saying is that they are carriers of the trait, so they're heterozygous, but they're not affected by it. Okay, so when it comes to gender is also decided by obviously the parents' genes, okay, because the parents, your genes are going to control everything about you, including what gender you are. Okay, and I know there's talk about, you know, what a sex is, what a gender is. When it comes to biological terms, there are 2. You do get hermaphrodites and you do get hermaphroditic types of animals, which are both genders or can switch genders. Okay, but in this biology we're going to just be looking at 2. So you are either going to have the XX chromosome, which makes you a female. You are then going to have female traits as well as the female reproductive organs, or your cells are going to have the XY chromosome, the male chromosome. Okay. Now, interestingly enough, because the female only has XX chromosome, it is actually the male sperm that is going to dictate the gender of the offspring, because the male sperm can either be X or it can be Y, whereas the female's egg can only be X. Okay, so to that king that killed his wives because they weren't giving him sons, he should have been the one on the other end of that. Anyway, so there's obviously going to be a 50% chance of being male or female, boy or girl. Okay. Now, some inherited disorders. Okay, so of course, if there is a mutation in your parents, the chance that you'll also be getting it. Something like polydactyly. Polydactyly means people that are born with another finger. Okay, or they're born with multiple toes. Okay, it's hereditary, it's a dominant allele too. So if your parents have it, chances of you having it are very high. Okay. Another one is cystic fibrosis, which is a cell membrane disorder. Results in thick layers in the airways, and the pancreas causes by a recessive allele. So both parents must have that. So both parents must be carriers of that recessive allele, or one of them be affected and the other one be a carrier for the children to then be affected, for the offspring to be affected. Okay. You also get sex-linked disorders. Okay, and what that means is disorders that are found on the X or Y chromosome. So there are some disorders that are only found on the Y chromosome, such as color blindness. Okay, if color blindness is passed down from father to son, okay, you do get female color blindness, I think, but it's mostly in males. And so if the father has color blindness, the son will have color blindness because that trait is only found on that Y chromosome. Okay. So once these gametes join together and are fertilized, we theoretically won't know exactly what disorders or what traits a fetus will have until they're born, and we can actually see them, okay, and we can see how they grow up. However, they have developed methods called embryonic screening, okay, and what this is they are going to analyze the cells of the fetus before they're even born. Now, there are three ways to do this. Amniocentesis. This is during quite late stages in the pregnancy. They stick a needle through the mother's stomach into the amniotic fluid, which is in the amniotic sac, okay, and they draw some of that fluid out. Now, some of the baby's cells are going to be in that amniotic fluid as well. They then can analyze those cells and they can see if there are any disorders with the child before they even come out. Okay. Another one is chorionic villus. Chorionic villus is the removal of some of the embryonic cells of the embryo. Okay, so they find the embryo and they remove some of the cells, but they're removing it, as they say here, between 10 and 12 weeks. Okay. Also by using the placenta tissue. Okay, same thing, they're going to look at those cells and make sure there's nothing wrong with the child. And then there's IVF, which is when this is in vitro fertilization. So this is when we were talking about the other day about them fertilizing the egg and the sperm cell outside of the mother. Okay, and then implanting only healthy embryos, fertilized eggs, into the mother. Okay. Now, this obviously has some ethical issues. If we're talking for embryonic screening, it reduces the number of people suffering from the disease. Okay. It's also very expensive to treat this disorder. Okay. There's some disorders that are going to lead to that person having to spend money for the rest of their lives to survive, or just to have a fulfilling life, okay? And there are also going to be regulations in place so that only parents that may have a history of this certain disorder being allowed to screen, and no unnecessary screening. Now, the arguments against the embryonic screening, however, it could discourage discrimination of parents that choose desired characteristics. Obviously, if a child does not have those characteristics, or if a child is affected by a certain disorder, parents could choose to abort the child. I say the child, the fetus, okay? Parents could choose to abort the fetus, which means they don't carry on with the pregnancy. When they see that their fetus may be afflicted with a certain disorder and have to live the rest of their lives with that disorder, they instead abort the fetus. Okay. And another one is saying that this might promote the idea that people without this disorder, or people with this disorder, won't be able to live fulfilling lives. Okay. This happens a lot with children that have Down syndrome. Down syndrome is something that can be detected during this chorionic villus or even the amniocentesis. Okay. Now, some people would rather abort the fetus than have the fetus grow up to have Down syndrome. Okay. But that's not to say that Down syndrome people can't live fulfilled lives and live full, long lives. You see. So ethical issues here. It's really up to the individuals and what their beliefs are. Okay. So, my name is Cameron. This has been genetics, and I will see you in the next lesson.